Welcome to our Biomed Unleashed podcast. I'm Alisa, a student specializing in regenerative medicine at Maastricht University.
And I'm Misha, also a student at Maastricht University studying the Maastricht Science program.
Our podcast is endorsed by the Maryland Institute for Technology-Inspired Regenerative Medicine and supervised by Professor Lorenzo Moroni. We aim to make regenerative medicine accessible and understandable to everyone.
In our episodes, we invite scientists, specialists, and innovators to share their career insights and latest discoveries in this exciting field of regenerative medicine.
Today, our guest is Professor David Schaffer, an important figure in the fields of chemical and biomolecular engineering.
David Schaffer has been a professor of chemical engineering, bioengineering, and cell and molecular biology at the University of California, Berkeley since 1999. His research focuses on modifying viruses for gene therapy purposes. Previously, he obtained his bachelor's degree in chemical engineering at Stanford University and PhD at MIT.
Professor Schaffer is also the director of the Berkeley Stem Cell Biological Engineering Training Program at the University of California, Berkeley. Moreover, Professor Schaffer is a co-founder of a number of biotech companies and ventures. Throughout his career, David Schaffer has won more than 20 awards. Hello, Professor Schaffer. We're Alisa and Misha, and we're very grateful to welcome you as our guest for Biomed Unleashed podcast today.
Good morning, Professor Schaffer. Yes, thank you so much for giving us such an incredible opportunity.
Terrific to be speaking with both of you. Thank you.
So you have had an outstanding career spanning multiple top institutions and groundbreaking research in gene therapy and stem cells. But if we go back to your childhood, you mentioned that your father, who was a biochemist, used to come up with different mnemonic techniques, which made it fun to learn and develop the natural interest in biology. Did you have any other role models or situations that sparked your interest to start your scientific journey in chemical engineering and biotechnology?
Sure, this is probably not too shocking, but yeah, my mother and father both role models for me. My father is a retired professor. He worked in a biochemistry and pharmacology department his entire life as an academic, researching, mechanisms of cardiac decline. My mother is an MD who took some time off to be able to raise the family, but then returned to work at what became Novartis and then what became Sanofi, the precursor companies that were merged into Novartis and Sanofi, and did drug development and was running phase 1 through phase 4 human clinical trials and gathering data to get FDA approvals of drugs. And a couple of her drugs are still on the market. So that, both of those folks really strongly influenced my career choices. And I probably ended up following a path that's, maybe exactly halfway in between the two of them, where I was academic, but more applied than my father, but not so applied as to be involved, directly in running the clinical trials with inventions that we hope to make.
And during your childhood and early education, did you face any significant challenges that possibly shaped your career in the future?
Nothing like highly unusual, I would say, in the sense that I grew up for a number of years in Eastern Pennsylvania, New Jersey, spent some time in the South in Alabama growing up as well. I would say that, different parts of the US have different cultures when it comes to, education and career development. And the South back in the 80s was a very different place. It's different from how it is today, but it's also different from the Northeast in that, progressing as a nerdy scientist and an engineer is something that is viewed differently in some states versus others. But nothing, I didn't have any significant hurdles, majorly different from many of my colleagues. And, got to a position when I started undergrad where I was not, thrilled to be able to have the opportunity to continue to grow and develop and move forward.
Okay, and if we now go to your academic path, you have obtained your bachelor's degree in chemical engineering in Stanford University and your PhD at MIT. You did your postdoc at the Salk Institute, eventually becoming a professor at the University of California, Berkeley. So could you please share at what point and why did you decide to work with genes and stem cells?
Yeah, I became very interested in chemical engineering because, as I mentioned earlier, I love biology, but I like being more on the applied side of biology. I like developing technologies. I like solving real world problems, and really gravitated towards therapeutics, in large part because of the influence of my mother. So if you put together, biology, technology development, engineering, that at that time, and still that's chemical engineering. Bioengineering has grown up largely as a parallel field to chemical engineering in the past couple of decades, but there weren't as many bio-E departments when I started out as an undergrad. So chemical engineering really became the career choice where I could blend together engineering of biomolecules for therapeutic application. A lot of chemical engineers were pursuing more traditional directions like the petrochemical industry, but there was already a very strong group of people working on biological applications of chemical engineering principles back when I started. So now, my home is, I have appointments in both chemical and biomolecular engineering. A lot of the chemical engineering departments have revised their name to contain something bio. So we are chemical and biomolecular engineering. But I also have an appointment in bioengineering as well as molecular and cell biology on the campus. So we really enjoy being at that interface. So undergrad was a terrific time to learn, the basic disciplines of biomolecular engineering and biology. Graduate school was the time where you start making a choice of career direction and research direction, certainly. And I've always been somebody who's gravitated towards risk. So calculated risk, not jumping off a cliff risk, but calculated risk. And at that time, if you ask, where was the really exciting innovation when it came to therapeutics and therapeutic modalities, obviously small molecules are very well established. Drug delivery was just something interesting, was becoming, pretty well established as well. Antibodies were relatively new. This is back in 1993. But those were, there were FDA approvals, and that was becoming somewhat established. And this new concept was floating around, relatively new, about instead of a small molecule or a protein as the therapeutic, DNA could potentially also serve as a medicine. And in its most straightforward form, gene therapy addresses the idea that if you have a broken gene, if gene X is broken in tissue Y, if you deliver gene X, a replacement copy of it, to tissue Y, you could treat a disease. And people were thinking about diseases such as cystic fibrosis or hemophilia. And there was a lot of enthusiasm within the field where DNA could be a medicine that could potentially be one and done, that if you deliver it one time, because DNA has the potential to persist for the life of a patient or a person, you could have a single administration, long-term therapeutic benefit. And the big problem, it was already apparent in the field, was delivery. How do you deliver that DNA to cells and tissues? Small molecules, because of their properties, tend to biodistribute through the body and cross membranes and get easier access to cells. DNA by comparison is a big molecule, and on its own, floating in the body, and at least in serum, is not particularly stable. So we needed delivery vehicles to protect the DNA and get it to the target site. And that has been a major challenge since the inception of the field, continues to be a significant challenge. But that was the challenge I decided to dive into for my graduate work, because you could see the transformational potential if you could deliver DNA efficiently.
And I think at that time it was unbelievable that it's possible to do that, right? Did you have this belief that you will be able to make it? Or
well, I think probably even I underestimated the magnitude of the delivery challenge. You could see, even then it was a pretty big challenge, but it's been, a long time coming to develop delivery vehicles that are efficient enough to start seeing efficacy. And now, at last count, if you count genome editing, Casgevy, it's a nucleic acid therapy. There are 18 FDA-approved gene therapies. So this has really been picking up a lot of steam, especially over the past 5 years or so. And all the promise that we saw, 30 years ago is really turning into potential and into therapeutics and into cures now.
wow. And do you see any connections between the work you do today in gene therapy and stem cell research and hobbies or activities that you did as a child?
Do you mean as a child as in an undergrad, or a child as in a 5-year-old?
I think a child, like 5, 10 years old, maybe.
Well, as you mentioned, my father was and is an educator through and through, and I do have, my sister and I have distinct memories of sitting on his lap while he opened up textbooks and was going through. I mean, his two favorite things were to teach us were anatomy and microbiology, because I think he was teaching these himself. And going through and memorizing parts of the body, bones and muscles, and memorizing on site. He'd show us a picture of a microorganism, and we had to know the name. So medicine and therapies or therapeutics are something that I was interested in from a very early age. I was not nearly savvy enough or sophisticated enough to think about gene therapy back then, but certainly the seed of wanting to develop therapies to treat human biology and human disease was something that was planted very early in life.
Okay. And the main area of your research is gene therapy. As we understand, you create viral vectors, which are modified viruses designed to deliver genetic material into cells to treat a disease. And for that, you use directed evolution. Can you please elaborate on the process of directed evolution for engineering viral vectors? What is it?
Sure. If I could take a step back and talk about why we use viruses for gene therapy, that'd be great. So as we mentioned, really the big challenge in the field, we have this joke in gene therapy. There are only three problems: delivery, delivery, and delivery. And so you need a delivery vehicle to get DNA inside of cells. And people, relatively early in the field turned to viruses because viruses deliver nucleic acid to cells as part of their replication cycle. That's what, part of what they do for a living. And perhaps we could take a virus, ideally a harmless one, and disable it and delete its DNA, its nucleic acid, and instead insert a DNA medicine, the gene of interest you would like to deliver to treat a disease. And then as a result, trick that virus into delivering your therapeutic payload rather than its own DNA. So we're, essentially using viruses as Trojan horses to carry in our payloads to treat disease. There are challenges associated with that, though, which include the fact that viruses are natural products of evolution, and we're trying to use them as biopharmaceutical delivery vehicles. And the natural properties that have evolved with many viruses are not compatible with, not efficient enough, not targeted enough for therapeutic gene delivery to treat human disease. So we have, some promising natural viruses. We've been working with this one in particular, as has the field. But I started working with this virus in the mid-1990s called adeno-associated virus. And it's currently the basis of 6 of the FDA approvals. So there's 6 FDA-approved gene therapies based upon AAV. And investigators in the early 90s showed that you could actually use it to deliver a recombinant molecule, a therapeutic cargo or a piece of heterologous DNA. But the big problem is that AAV is a pretty harmless virus in nature. It's been evolving for tens of millions of years, and the majority of the human population has been exposed to it and never even noticed it because it's not associated with human disease. So it's a good starting point, but it's also kind of a mild respiratory virus. It's not that very infectious on lung even. And if you start showing it other cell types or other tissues that it didn't even involve to infect, such as skeletal muscle or cardiac muscle or the brain or the retina, it's very ill prepared by evolution to infect those cell types. It's different from the properties that nature endowed it with. So what that to us meant is that to make very efficient targeted gene delivery vehicles based on this or other viruses, we needed to re-engineer those molecules, at the amino acid level, those viruses, to make them different and reprogram them to be highly efficient at mediating DNA delivery to, a neuron in the eye or in the brain or a muscle cell in the heart or in the skeletal muscle. And that was a very difficult thing to do. AAV, it turns out, is about a 5 million molecular weight molecule. We think it's challenging to re-engineer antibodies. Really, what you're engineering is the tips of the antibody, the region that confers specificity. That is really small compared to a 5 million molecular weight AAV particle that contains nucleic acid and DNA, nucleic acid and protein. So in addition, from an antibody, you're asking it to bind something. With AAV, we're asking it to biodistribute through the body, interact with lots of bodily fluids, find the target cell, bind to it, get internalized, escape into the cytosol of the cell, traffic to the nucleus, and then dissociate the DNA from the protein to release the DNA cargo. So we're asking a lot of this cargo, of this vehicle. It's a very complex process. So we did not know back in the 1990s and don't know today enough basic molecular information, mechanistic knowledge, in order to engineer that particle rationally, to design the surface of that particle to mediate specific delivery to neurons, for example. So we turned to another approach and recognized this idea that this virus, as I mentioned earlier, has been evolving for tens of millions of years. Why not allow it to continue to evolve, but do that in the laboratory in a directed way so that you harness evolution to empower this virus to specifically and efficiently target different cells from what it evolved to infect. Engineer new versions that excel at infecting neurons, for example. So this approach of using evolution as a design tool was actually invented in the early 1990s by a brilliant investigator by the name of Frances Arnold. She got her PhD at UC Berkeley, started up a career at Caltech, and invented this concept of using evolution as a design tool in her work with industrial enzymes, with enzyme activity. And rather than staring at a crystal structure and deciding which amino acids to change, she started creating large libraries of randomly mutagenized enzymes and then conducting a high throughput screen to identify the fittest, and then taking the fittest, re-mutagenizing it, rescreening it for the best in that next round, and performing these iterative rounds of genetic diversification and selection that emulates the natural process of evolution, but in the laboratory in an accelerated way to fundamentally change the properties of enzymes in useful and desirable ways, to make them more thermostable, to make them catalyze different reactions. So that work was initiated in the early 90s. I started up my lab in the late 90s, became an admirer of that work, and we were the first to start applying it towards AAV and towards gene therapy, where we started genetically diversifying the coat proteins of the virus, because that capsid, that coat protein of the virus, is the delivery vehicle. And we started creating large variant libraries and then selecting them for the capacity to better deliver DNA to a target of interest, whether it be cells in the retina, the brain, the heart, the lung, for example. So fast forward 25 years, we started doing this when I opened up my lab in 1999 at UC Berkeley. And we're thrilled that technology is now advanced into non-human clinical trials and is generating positive data with variants, novel viral variants that we created.
And you mentioned regarding the viruses and when you introduce them to the body, that you remove the DNA of the virus itself and then you insert the needed DNA that you want to deliver. But I think that anything that gets introduced to the body will cause an immune response. So is there an immune response when you introduce the even edited, like engineered virus, or how does it work?
Yes. So the immune system, I mentioned a little bit earlier that, even I or the field didn't recognize the magnitude of the delivery challenge that we would be facing. When I entered in the gene therapy field in 1993, one of the big challenges has been interactions with the human immune system. And there are a couple of ways in which this happens. There's what happened before the dosing with the gene therapy, and then there's what happens afterwards. So before, I mentioned AAV is naturally a human virus. So a lot of us have already been exposed to it. And a lot of us have been vaccinated as a result, accidentally, through natural exposure to natural AAVs. So a lot of us have high concentrations of antibodies directed against these natural AAVs. And if we are administering the virus through route of administration, such as injection into the bloodstream, that exposes them to these antibodies, then the antibodies neutralize the virus and neutralize the therapy. So we've been trying to tackle this. There's several potential solutions in the field. Our approach has been to evolve versions of the virus that are resistant to pre-existing antibodies in the human population. So that virus would be new to a patient, new to you. And so we've created one that's actually being used in a human clinical trial for cystic fibrosis, which I'm happy to talk about more. That's being run by a company I co-founded based on my work. But that variant that's being dosed in patients with cystic fibrosis is a virus that is substantially more resistant to pre-existing antibodies in the human population since we evolved it to be resistant to those antibodies. So that's what happens before the administration. Afterwards, there is a phase where the body recognizes that there's foreign virus that's present, and it begins to mount an immune response against it. And typically, we in the field immunosuppress the patient. That's just standard of care now, that you do immunosuppression with a steroid, something like prednisone, at the time of administering the AAV, so that the body doesn't overreact to this foreign nucleic acid. And provided that the doses of AAV that are used are reasonable, AAV is a pretty stealthy virus. If you exceed a certain amount, the body's really going to notice it and react badly against it. But as long as you're staying below that amount, which is easier for us to do because we're creating more efficient variants where we can go in with lower doses, then that immunosuppression works, that transient immunosuppression. And then you taper somebody off of that immunosuppression so that they're no longer receiving prednisone. And then in the longer term, though, you can temporarily suppress the immune system, but it's still surveilling enough that it actually notices this foreign coat protein of the virus and raises antibodies against it. So patients typically, after they've been administered with the AAV, get really high titers of antibodies, concentrations of antibodies directed against that new AAV. So it would be hard to re-administer the AAV because it was new to you the first time. The second time around, your body's seen that before. You have antibodies against it. But fortunately, for most of the applications we're envisioning gene therapy and AAV for, it's one and done. That you'd need to deliver that DNA once, and then once the DNA has arrived safely at the target site, it persists for the life of the patient, and you don't need to redose with virus.
Okay, and your work also significantly impacts stem cell research, particularly research in understanding and controlling stem cells' fate decisions. So, and how have novel technologies that were developed in your lab improved our understanding of stem cell behavior?
Sure. We've worked with gene therapy, genome editing, and cell therapy. The common thread through those is that we view all three of them as having DNA as their therapeutic modality. That if you're adding new DNA to a genome, that's a gene therapy. If you're editing existing DNA in the genome, that's an edit. If you're adding an entirely new genome to a tissue or to an organ, that's a cell therapy. But the common thread, as I mentioned, is that all three of the quote unquote active ingredients is DNA. And the advantage of DNA, again, is one and done, that a single administration has the potential to provide a lifetime of benefit. So for each one of these, being an engineer, I want to identify what is the big bottleneck and try to engineer technologies to alleviate and overcome that bottleneck. In the case of gene therapy, the big initial problem has been delivery. Now that we're starting to see, light at the end of the tunnel, that as a field, we're creating solutions to the delivery problem. There are new problems that are coming along, such as how do you manufacture enough virus? But in the case of stem cells, the big challenge is how do you control them? That a stem cell is an immature cell that has the capacity to continue to divide in an immature state, to self-renew, as well as to specialize or differentiate into one or more cell types. And typically, we need them to specialize or differentiate into a population of cells that is missing in the body due to some disease that kills off populations of cells. So think Parkinson's disease, which is due to the loss of midbrain dopamine neurons, type 1 diabetes, which is due to the loss of beta cells. Once an entire cell population is lost, you can't really use a gene therapy to repair the tissue. You need to repopulate the tissue with new cells. And so that's where stem cells come in. The big challenge, though, is how do you control the stem cell? How do you take that blank slate? How do you take that self-renewing, dividing immature cell and control its differentiation into the specific cell type that you need to inject to rebuild a tissue and restore function to that tissue? So stem cell control is the big challenge in that case. And there are two parts to that. One is, what are the natural biological mechanisms that control self-renewal and differentiation for a stem cell? And second, how do you scale that in a bioreactor, for example, to the point where you can treat thousands or tens of thousands or even millions of patients with some of these conditions? So that's the problem that we've been working on since my lab opened in '99.
Mm-hmm.
And it also takes a lot of time to culture the cells to make them differentiate. How do you work with that? Because, for example, to treat the patients, sometimes you don't have a lot of time to do that. So
yes, it depends on the condition, and so we have been starting with conditions where there is a therapeutic window. Somebody's been diagnosed with Parkinson's, for example. They start taking L-DOPA, which I'm sure you're familiar with. That's able to restore some of the dopamine function within the brain. Eventually, L-DOPA fails. L-DOPA is a treatment. It's not a cure. It does not modify the timeline for progression of the disease. It simply importantly, alleviates symptoms for a period of time. But eventually, patients lose the dopaminergic function, and that's when they become, strong candidates for a cell replacement therapy to repopulate the brain with those dopamine neurons that have been lost, that have died due to Parkinson's disease. So you have a window there. Type 1 diabetes is another example that patients start getting an immune response against their own beta cells. And as a result, the beta cells are lost, typically in childhood. They then become candidates for insulin replacement therapy, which is a treatment. It's not a cure. Patients still suffer from a number of the effects of diabetes. So insulin injections are a partial fix, but it would be terrific to repopulate the beta cells in the body. But again, there's a window of time where you can do that. Because patients are enjoying the benefits of the insulin therapy for a time before they potentially can become terrific candidates for cell replacement therapy. So our goal in both of those cases is to develop scalable, economical, high quality, reproducible processes to generate large numbers of cells to treat those patient populations.
And there are currently different types of stem cells used in biomedical engineering. So what types of stem cells do you use in your research?
We work with pluripotent stem cells. So there are, two buckets of stem cells. A pluripotent stem cell is, something that's reminiscent of what was present within an early embryo, but these days you can create these cells in a number of different ways that don't involve embryos. But these pluripotent stem cells have the capacity to differentiate, have the potential to turn into every cell in the adult body. So from that single cell, you could generate cell replacement therapies for a pretty broad range of conditions. Then there are adult stem cells or multipotent stem cells that have undergone some specialization. So hematopoietic stem cells, blood stem cells have the ability to turn into everything in our bloodstream, a lot of red blood cells, all the immune cells. And as a result, you could use them to treat conditions that specifically affect the blood system. And there are, strong therapies based upon hematopoietic stem cells, or bone marrow replacements. That's the active ingredient there is the blood stem cell. So both pluripotent stem cells as well as some multipotent stem cells have a lot of potential to treat a broad range of diseases. My own research has focused primarily on the pluripotent stem cells, especially of late, where we would love to be able to use these cells as a single universal source to differentiate into a broad range of cell replacement therapies to treat a number of diseases.
Mm-hmm. And you also mentioned that the different stem cells could be derived from different things. And where do you source the stem cells from?
There are two major sources the field uses for pluripotent stem cells. Well, typically for adult stem cells, it's often autologous, that you're harvesting somebody's own blood stem cells, maybe doing a gene therapy on them and returning them to the body. So there are FDA-approved gene therapies like that. So this is a merger of two fields, right? It's part stem cell therapy, part gene therapy, where you harvest the blood stem cells, you deliver DNA to them to correct a genetic mutation present within those cells, and then you return them to the body. So in many cases, for adult stem cells, the source is autologous. It's somebody's own, own tissue. In the case of pluripotent stem cells, there are two major sources. One is these were originally derived from embryonic stem cells. And the idea here is that you're familiar with in vitro fertilization. It's been around 40-ish years. And in this, in this technology, there's a couple of us having a hard time having children. So you harvest eggs, you fertilize them in vitro, you create, a number of embryos. And then typically you take a couple of those embryos, implant them into the mother. And, if things go well, then, the couple is able to have a child. And incidentally, because you're implanting multiple embryos, there's a higher incidence of having twins in vitro fertilization, which you're probably familiar with as well. But you don't typically use all the embryos, and they're leftover ones that are frozen in the fertility clinics across the country. And estimates indicate that there are over 400,000 frozen embryos across the country. So the IVF kids are now, in their 40s, which means the IVF parents are way beyond the age where they want to have kids. And the question becomes, what do you do with those leftover embryos? So in some cases, those embryos have been donated to science and to medicine. They would have been destroyed anyways, but they're taken, they're used to derive embryonic stem cells, which is this, the first major source of pluripotent stem cells. And then there came a Nobel Prize for pluripotency reprogramming. So Shinya Yamanaka from Japan discovered that if you take any adult stem cell and you overproduce certain proteins, transcription factors that are characteristic of and very important within embryonic stem cells or pluripotent stem cells, you can turn back the clock in that adult cell and reprogram it into an embryonic-like stem cell. So those are called induced pluripotent stem cells. So human embryonic stem cells and human induced pluripotent stem cells are functionally quite close to each other. So they're used almost interchangeably now for cell replacement therapy applications.
And you talked about different projects with stem cells, like to treat Parkinson's disease. Are there any other projects or technologies that use stem cells that you're particularly interested in right now that are maybe promising?
I think there are a number. If you take a look at Parkinson's, type 1 diabetes, some forms of epilepsy, potentially some immunostimulatory strategies to treat cancer, I think stem cells are incredibly promising. One challenge that we've been working with, or one technology that's the basis of a company that I've co-founded, is how to scalably generate all the cells that are needed to treat these patient populations. And we refer to it as the football field problem, that if you take, let's say you're trying to treat enough dopaminergic neurons to treat the Parkinson's population, and typically these cells are cultivated as adherent cells on two-dimensional surfaces. Naturally they grow within a soft three-dimensional tissue, but the culture systems that are used as the standard within industry right now to grow up these cells involves adhering them to a two-dimensional surface. If you do the math, we're going to need, whether it's for Parkinson's or congestive heart failure or type 1 diabetes, anywhere from tens to hundreds to thousands of football fields worth of surface area to generate enough cells to treat the patient populations of these various conditions. So in addition, the culture systems that involve two-dimensional adherent growth have to end with removing the cell from that 2D surface so you can harvest it from the bioreactor and implant it into a patient. That removal step involves some pretty harsh treatments, enzymatic treatments, mechanical treatments that actually damage the cell. So end up with a problem that with the current processes, the quality or the health of the cell is not as good as it should be. So we decided to invent a three-dimensional process for growing, expanding, and differentiating stem cells. And by moving into that third dimension, all of a sudden the density of cells you can achieve is much higher. So you can work with a small three-dimensional bioreactor rather than working with these sheets, these bioreactors that involve sheets of cells, which are just much, much larger and more expensive. And in addition, if we can grow it in 3D, it's already in a state where you could administer it through a syringe into the body, just gently dissociate the cells from 3D and then administer them to a patient. So the secret sauce behind being able to do this three-dimensional culture is that we embed cells inside of a polymer, inside of a hydrogel, kind of the consistency of a contact lens. But we make beads of this hydrogel and we grow the cells inside these beads inside of a bioreactor where the beads are kind of spinning around in 3D with mixing from the bioreactor. And then we can release the cells from these beads very, very gently by using a change in temperature. And as a result, you can grow them up at 37 degrees at incredibly high densities under fully defined conditions, very reproducible process. And you can differentiate them inside of these beads of hydrogel, of polymer. And by changing the culture medium and adding differentiation cues. And then when you want to harvest them, all you do is drop the temperature. This polymer turns back into a liquid from a gel. And then you can very easily recover the cells using a very gentle process. And it's enormously high numbers of cells by virtue of the fact that we have these high density 3D bioreactors. So we're currently advancing that technology towards the clinic for a couple of targets, and fingers crossed it'll enter into the clinic within 2 years.
Wow. Oh, this is incredible. Yeah, during my university studies, I learned, because I studied regenerative medicine, and I learned this, and now you're telling me that it's maybe now possible, and I'm like, wow. Also regarding 2D cell cultures and 3D cell cultures, and also testing the drugs or maybe also gene therapies, we want to ask you a question because we know about the organ-on-a-chip systems, which are microfluidic devices that simulate functions of human organs on a small chip. And the system can replicate physiological and mechanical functions of tissues or organs, and they provide a powerful platform for drug testing and disease modeling. And do you use them in your research to test gene therapies or something?
We use human tissue. We don't use organ on a chip per se. And my view is that before going into the clinic, it's best to test a therapy or develop a therapy on the best available preclinical system so that you show efficacy, prior to going into human. It just maximizes the chances that it works. So some combination of the best available animal model, such as a large animal model, as well as the best available source of a human tissue is, to my mind, the best approach towards de-risking a technology prior to initiating a clinical trial. So that raises the question then, what is the best available human tissue? And in some cases, you can access primary human tissue and culture it. In other cases, taking a stem cell and turning it into a mini organoid on a chip is a good approach. So we have worked with situations where we're capable of getting primary tissue. So right now we're developing delivery vehicles for the brain, and we have a collaboration with neuroscientists where we can actually get access to primary human neurotissue. So we're using that as a tool. But in other situations, we've actually taken stem cells and differentiated them into cultures because that was the best source of that human cell type in order to test the therapeutic prior to going into the clinic.
Professor Schaffer, and your lab has made many groundbreaking discoveries, and sometimes they come from unexpected results, referred to as the Eureka effect. And do you have any surprising or unexpected discovery in your lab that led to a major breakthrough?
Well, yes, we have. I mean, there have been situations where we got lemons and tried to make lemonade out of them. This will take probably a 30-second intro just to motivate the problem. But we, in addition to creating scalable systems for manufacturing therapies out of stem cells, we're interested in that initial biological problem, which is what are the signals that tell a stem cell to divide versus differentiate? And so we developed a strong interest in studying signals and manipulating and controlling signals within stem cells. And to discover at a very basic level the kinds of signals that are involved and the dynamics of those signals, we wanted to develop a technology based on optogenetics, where you shine light on a cell and it activates particular signaling pathways. And as a result, you can vary, the very tight control activate particular signaling pathways that might play a role in stem cells division or differentiation. And you can study the relationship between the input signal and the output cell behavior. So we were generating or engineering optogenetic tools, which are proteins that take in a photon, light, and manipulate a signaling pathway. And one of them worked completely opposite to what we thought it should, that we wanted a molecule that dimerized, and it created these large oligomers, these large aggregates inside of cells. And that was very surprising to us and initially very disappointing to us. And terrific graduate student in our lab named Lucas Bugai, who's a professor now of the PI of his own lab at the University of Pennsylvania, recognized this as an opportunity. We thought it was a failed experiment. And he began to recognize that during the activation of many signaling pathways, there are protein intermediates to these pathways that self-oligomerize into larger structures as a necessary and sufficient step for the activation of that pathway. So we started taking these proteins and fusing them to our optogenetic protein. So if you shine light, all of a sudden you created a oligomer of the signaling protein, and the cell, then activated the signaling pathway downstream of that. So it was a complete accident, something that we didn't expect, thought it was a failed experiment, and turned into a new line of investigation that's led to about 10 publications so far.
Mm-hmm. Oh, that's so cool.
Yeah. And so if we go back to translation processes, as you told us, many technologies from your lab are already in human clinical trials, and you also have co-founded 8 companies based on your research. And how has your experience in founding and leading biotech companies influenced your approach to academic research?
Yes, I've always been really interested in that interface between academia and industry, probably dating back to my admiration for my parents' careers, where my father worked in academia, but very much on the basic side. My mom worked in late stage drug development, and I've really been interested in that interface. And that interface has been, people refer to it in some cases as the valley of death, that it's really hard to get a technology and invention from an academic lab onto a path towards a clinical trial and a successful product that is good for patients, that society can benefit from. And there are a number of little valleys of death in between. There are a number of challenges you have to overcome for that translation process. And I've had a lot of fun working very hard, both initially on the academic side, and then putting a lot of effort into the companies to enable us to translate technologies and get them into human clinical development. As I mentioned to you, I'm very thrilled that we've done this a number of times, and my companies are now running a good number of human clinical trials. Your question is then, how does that feedback and affect my academic research? And I'm going to say this in a way that exaggerates the problem. You want to be sure that as an academic developing technologies, you're trying to solve a problem that actually exists. Meaning that we've always started with and increasingly so, because my connections and ties with medicine have grown over time, and with industry have grown over time. You really want to be sure that if your goal is to solve a societal problem, it's actually a problem. That if you developed a technology that had a certain property, that physicians would use it, and the medical system would use it. And sometimes, if you don't know that, the needs of those quote unquote end customers, the physicians, and of course, the really important patients they're treating, you might go off in the wrong direction and design a technology that's not suited for a given application or for clinical application at all. So we always start with the end problem in mind, and then work our way backwards to a strong technological solution for that problem, or ideally for a set of problems. We tend to develop technology platforms rather than single solutions for specific problems. So that's one strong piece of advice I always give people, that there is a lot of room for basic discovery in academia, where you're studying interesting problems and you want to discover something new about biology. And that led to the discovery of RNAi. It led to the discovery of CRISPR. But if you're at that interface, if you're an engineer who's trying to solve specific problems, really learn a lot about the problem, talk to the physicians, talk to people in industry, and start with that problem in mind prior to trying to engineer the solution. So I think we've gotten a lot better at picking problems and picking solutions as a result.
So can I say, in other words, be more realistic in this sense?
I think more aware of the full context for the challenge you've decided to take on. So speak with the physicians, speak with the drug developers, and design something that's really going to solve the problem that's the focus.
Okay, and could you share some specific examples or of challenges overcome and lessons you learned during this translation process, if you had some?
Sure. Pick the right drug target. As I mentioned, we develop technology platforms, and it's a platform technology is one that can create products that address a set of potential problems, disease indications. And it's really important to point that discovery engine in the right direction so that you're creating your first product, which is, maximally likely to succeed. And so you want a product where it's the least risky path towards at least moderately successful commercial product. It doesn't have to be a blockbuster the first time around. If the biological risk is higher to get that blockbuster. But it should be the least risky path towards showing success in the clinic for something that is a reasonable market. And then once you've de-risked the technology, you can then start taking on more biological and medical risk and going after tougher targets. But yeah, my first company, we picked the wrong clinical target as the very first one, probably lost 2 years as a result of that. And we learned the company's still around, and we're after what's a really good target right now. But it's important to get that decision right from the very beginning. Get to know and trust the people you're working with incredibly well. That a co-founding team of co-founders, of investigators, are going to be working very closely together for a number of years. And it's really important that there is, a very strong common sense of purpose and trust amongst that team. So get to know people really well before, pulling the trigger of deciding to commit or work together. That's another, I think, challenge that has gone both ways. Ideally, if I've made a mistake once, I never make it again, which I think is the case. And fortunately, none of these mistakes have been fatal. These companies are still growing, have either been acquired, gone public, or growing. But it's great. I want to spare other people the fate of making some of the same mistakes I may have made.
And if we talk a bit more about your mentorship, you have advised over 90 graduate students and postdoctoral fellows. Can you share a memorable mentorship story where your guidance significantly impacted a student's career?
I would say that there have probably been a number of them. And the way I approach mentorship is about happiness in life. That my goal is not to guide somebody into being a professor, or guide them into working as a scientist in industry, or telling them they should become a patent attorney. My goal is to ask a lot of questions to discover what makes them happy, because that's the goal, right? There's this saying from Mark Twain that if you choose your career right, you'll never work another day in your life. It should be about something that's joyful and enjoyable and fulfilling. So my view is that if you take just drug discovery, there's a lot of steps along the way. There's the basic initial discovery, there's the technology translation and towards industry and the industrialization of that. There's the development of a product, a clinical product based on that technology. There's, the getting it past FDA muster, getting it to clinical development, commercializing it, and getting it to as many patients as possible. That is a long and very complex process with lots of moving parts, and we need really smart people at every step along the way, including not just the drug developers, but the patent attorneys that are going to be protecting the IP. So my goal is to really discover what makes somebody happy and help them discover that for themselves, so that they end up finding the right place in that spectrum of activity for themselves. So I think I've done this well in a number of cases. I've had incredibly successful people who are 27 people from my lab are now have gone into academia, and most of them, they're either young professors, or the vast majority of them have gotten tenure. So In addition, probably the majority of people from my lab have gone into industry at various stages along the way. I have, former graduate students and postdocs who are CEOs and CSOs and CTOs of companies, including public companies, which I'm thrilled about. And there are folks who've gone into patent law, management consulting, education policy, scientific policy. There are people who are in Washington advising Congress people. So I guess, I can't think of a single moment that stands out any more than many others. But, I'd like to think that hopefully I've touched these folks' lives in various ways that, have led them onto a path to be very fulfilled and happy with their career choices.
Professor Schaffer, and our last question for today. So what would you do if not biomedicine?
That's an interesting question. I think, if you ask the question, again, I'm problem motivated. If you ask the question, what are the big problems facing our society right now? I think two big ones are human health and climate change. And there's overlap between those two because climate change is and will be increasingly affecting human health. So that's another area in which a lot of chemical engineers go, and it's a really important area. So I'll mention that part of my job at Berkeley now is that I direct a biotech incubator. We have 30 tenant companies, and we've developed a broad range of programs to accelerate these companies and enable them to succeed. And we're opening up a second incubator. It's actually almost under construction, and it's going to be focused on climate technology, and I'll be director of that as well. So I don't have companies that work on climate tech. It's not directly my research, but the idea is that we know how to run an incubator. We run it really well, and we'd like to extend that to another major problem, which is climate technology. So if I were to restart my career, 40 years ago and say, hey, I want to go a different direction this time, it would be towards climate change.
Okay. Professor Schaffer, thank you very much for sharing your exciting journey and your experiences with us. And we're truly thankful for your time and the opportunity to talk to you today.
Yeah, thank you very much.
A lot of fun to talk to both of you. I really appreciate it. Thank you very much. Very much enjoyed it.
Thank you so much for listening. We hope you enjoyed this episode of Biomed Unleashed podcast and will stay with us for the next ones.